In this video, we will discuss quantitative PCR, otherwise known as qPCR. Let's start by talking about the basic theory behind PCR. So quantitative PCR, otherwise known, as we said, as qPCR, is a method that allows you to quantitatively measure the mRNA being expressed in a cell. So to really understand this, let's start by going back to understanding nucleic acids. And I imagine that this is something everyone is very familiar with, but just to be absolutely sure, let's go through it one more time. So as you all know, there are nitrogenous bases that are the basis of our DNA. And these bases are adenine, thymine, cytosine, and guanine. You can use the mnemonic cut1pi to remember which ones are pyrimidines and which ones are purines. Those four bases are going to come together to form DNA. And this is the genomic material in our cells. And as you all know, every cell will have the same genomic material. And it will be these bases paired to form DNA, which lives in the nucleus of the cell. This DNA will then be transcribed to form mRNA. And this is the part where things get different. So up till here, everything is sort of the same in every single cell. But here, the cell will only transcribe the mRNA for genes that it needs. So this is based on cell needs. And this could be due to microenvironmental pressures, it could be due to changes in metabolism, it could be due to any sort of signaling that the cell is receiving from the environment, or it could be due to the function that cell normally performs. And so, for example, heart cells might produce certain things, whereas skin cells might produce other things. And so mRNA is sort of your measure of what genes are actually being transcribed, or what genes does this cell care about, and what genes does this cell need. And so, like we said, this is going to change. Now, mRNA is going to get translated to protein, and protein is, of course, your actual effector. So this is what can actually make change and perform functions in the cell. And so this is your true measure of what the cell is upregulating and what it is doing in any given moment. It is important to realize that although we are taught that mRNA is translated to protein fully, this is not entirely true. mRNA can sometimes be degraded. It can also simply be kept around until the cell actually needs that protein, so it can be sort of sequestered in place until it's necessary. So it's not always going to go to protein. And the importance of understanding that is understanding that mRNA is a good measure of what genes might be transcribed, but it is by no means a perfect measure of what genes the cell is actually using in any given time. To really understand that, we have to understand the protein. But that said, mRNA is a good and easy measure of what is going on. And sometimes our question is more about what genes are being activated and not so much about what proteins are actually being used in the cell. And so for those types of questions, mRNA is an especially useful tool. The other thing to know about mRNA is that sometimes in the lab, we will reverse engineer mRNA to the cDNA that it came from. So C here stands for complementary. And what this means is that mRNA cannot be used for every application. And so sometimes we need a DNA construct that we can then use for future applications. And so because mRNA has a slightly different structure. And so when we need cDNA, we can reverse engineer it in the lab using an enzyme called reverse transcriptase. And this will essentially take the mRNA and convert it back to the DNA that it came from. But it is important to realize that this is not the same as the genomic DNA. So cDNA is not going to be equal to the actual DNA here that we talked about, because remember, this DNA is the entire genomic DNA, including the parts that are not transcribed. So cDNA will only include those parts of the DNA that were transcribed into mRNA. So in the sense, it is the DNA of only the expressed genes in the cell. The reason I'm emphasizing these concepts so much is because, as we just said, qPCR measures mRNA, and that is why we talked about the utility of looking at mRNA. And it measures it by first converting it to cDNA and then performing the reaction. And that's why it is important to understand that cDNA is not actually genomic DNA, it's only the expressed DNA. So now that we understand those basic concepts, we can move on to talking about qPCR and how it actually happens. So normally when you are contemplating doing a qPCR, you will have 4 major phases to your experiment. The first one will be to set up your conditions of interest. So this will basically be get your cells or your tissue or whatever you're interested in, perform whatever conditions that you're interested in assessing, and then get ready for the next phase. The next phase will be to actually isolate the mRNA from your sample. And for this, there are a few methods, the easiest and most popular of which is probably to use the Qiagen kits in order to easily and quickly isolate mRNA. You will then convert the mRNA that you isolated into cDNA because, as we talked about, for some applications, including qPCR, you need a DNA sample and not an RNA sample in order to proceed. Once you have that cDNA, then you will be ready to actually perform the qPCR reaction, which, as many of you know, is a 40-cycle reaction where the amplification of your genes of interest are measured. So we will go through each of these phases in more detail now. Let's start with our experimental setup and mRNA isolation. So as we talked about, you will choose your model. This could be cells or tissues or animals. You will perform your condition. And as we've talked about in the past, these are some examples of conditions that you might use, but these are not the only ones. Don't be limited by these conditions. You can definitely do additional things like comparing different cell lines, perhaps comparing controls and knockouts or overexpressions, comparing other drug treatments. There really is no end to the types of conditions you can try. The only important thing to remember is that you must have an appropriate control for every single condition treatment that you do. And then from all of those samples, you will extract mRNA. Another important consideration here, which of course is a consideration in almost every experiment, is that these conditions should always be done in triplicate in order to get the most reliable results. We will then perform the reaction to generate cDNA from mRNA. And it is important to understand this reaction in theory just so that you understand what is happening when you perform this reaction. It's very easy to do in the lab, and probably the most important components of it are having this reverse transcriptase. So this is the enzyme that will actually go in and be able to regenerate DNA from your mRNA. But of course, to do that, it needs some supporting components, and so it will require your RNA template. It will also require extra primers, and these are random primers. So these are primers that will randomly align to the mRNA, and they will just provide a backbone for your reverse transcriptase to then build from. You will, of course, need extra bases, and so these are just all your bases available so that we can build DNA. You'll need a little water for the reaction to occur, and your buffer will usually contain some safety reagents like RNase inhibitors and DTT, which simply prevent degradation of the genomic material you're creating. So once you've mixed all these components in an Eppendorf tube, you will put them into a thermal cycler, and this is the reaction that will occur. And essentially in this reaction, you will have random annealing of your primers, because remember, these are random primers. And then your reverse transcriptase will come in and will sort of extend the chain and synthesize the rest of the DNA from the extra base pairs provided. And then finally, there will be a deactivation phase where we take out this reverse transcriptase, and this becomes a final strand of DNA. So now we've gone through sort of our setup and we've gone through our cDNA reaction. So now we're ready to actually run the qPCR. And we will go through exactly how to do this in much more detail in the practical side of this lecture. But I want to give you a basic list of what needs to go into this qPCR reaction. So just like we understood our cDNA reaction, it's going to be important to understand the qPCR reaction as well so that we can know what needs to be ready. So in every well of a qPCR plate, you need to make sure that you have cDNA templates and an appropriate amount of cDNA templates. You need to make sure that you have primers for your gene of interest. And please note that you can only use one primer per gene of interest. Unless you have primers that are specifically conjugated to different fluorophores, then you may consider using two. But in most cases, the primers you order will be non-conjugated primers that will be used for one gene. And so in every single well, please only include one set of primers for one gene of interest. You will then add in your qPCR enzyme mix. And so these are mixes that come commercially. An example is Cybergreen, which contains all the necessary components like our extra base pairs and DTT and all the other things that we might need, and contains a fluorophore, in this case Cyber, that the machine can see and can detect. And so that is how it will know how much your gene is amplifying. And then finally, you add in water to fill in the remaining volume. So this is sort of your basic template, and we'll go through the exact amounts and exactly how to set this up in future slides. So when we actually set up this reaction and we add in our cDNA and we add in our primers, it is important to understand what is actually happening. So as you know, we've put in our DNA sample, and that here is in green. And we've added in extra nucleotides as part of our mix. And we've also added in a primer for the gene of interest. Now, the reason for the primer is because remember that you took your sample. So these were your cells of interest. And you took all of the mRNA in that sample and you put it in a tube. So this tube contains every single mRNA in the entire sample. It's not just the one mRNA that you're interested in. And then you generated cDNA, which again contains every single piece of cDNA that would have been in that cell. And so you really need to tell your sample or tell your experiment what you're interested in looking at. Like, which gene do I actually care about? You're not measuring all of them in this experiment. You're only measuring one or two or maybe five that you're interested in. And so that is the role of the primer. The primer targets and isolates specifically the gene that you are interested in because only that gene can be amplified. If there are no primers, then that gene cannot be amplified. So the primer is what allows amplification of the specific gene that we're interested in. And because it gets amplified with these specially tagged nucleotides, the machine can read out how much amplification is happening. And to go back to some basic biology, the reason the primer is necessary is because our DNA polymerase can only build if there is a primer segment. It cannot build on DNA without a primer. And so if we add in that primer, we limit DNA amplification specifically to the part that we are interested in. So our primer here is going to be specific to this segment of DNA. We'll call this gene 1 that we are interested in. And it's basically going to go through a false replication cycle driven by the temperatures. So first you're going to denature it and separate it out. The primers are going to go in and bind. And then they're going to extend a new DNA segment. And then they will have created 2 new DNA segments. So from this one segment, we will have 2. And then we'll go through another set. And then we will have 4. And then you're going to go on and on and on and do this 40 times. So we'll generate 2 to the 40th copies of the gene of interest. And like we said, the reason we amplify like this is partially so that we can isolate the gene that we actually care about from this sample that has every single cDNA, and partially to create enough of a signal that we can actually measure meaningful differences between expression in the genes. Because if we have just 1 copy versus 2 copies of a gene, it's really hard to measure that in a meaningful way. But now if this is 2 to the 40th and 2 times 2 to the 40th, that is a difference that is detectable and measurable that we can then talk about. So to review this slide briefly, primers isolate the gene that you're interested in, and then they allow amplification. Amplification occurs over 40 cycles to generate 2 to the 40th copies of the gene of interest, and then measurement is performed during that amplification to determine how much amplification is occurring. And of course, if you have more DNA in your starting sample, then you will have more in your amplified sample, and so that's how we measure differences in expression. So now that we understand all of the theory behind qPCR, which is essentially understanding the mRNA and the cDNA and why we use primers, let's talk about the practical aspects of actually doing a qPCR. So this time we're going to step through, and it's going to be much more about what reagents, what volumes, recipes, how do you actually do this in the lab. So if you want to run a qPCR, there are some key preparation steps. The first is that you prepare your samples, and we're not going to go through this because this is about culturing tissues or taking care of mice. This is something we've talked about before. So you will prepare your samples, you will perform your treatments, and then you will extract mRNA. And mRNA can be extracted using any standard protocol. There are many protocols, and it needs to be stored in the -80 until you are ready to use it. mRNA needs to be kept very, very cold if you're going to store it for a long amount of time, so do keep that in mind. And once you make it, you can store it for a very long time, and you can use that sample to keep regenerating cDNA and keep doing more and more assays. So it is useful to make your mRNA and store it well, so then you don't have to do this whole step again of making all the samples. And then this should be done, of course, in triplicate, as we said. You will then generate cDNA, again using standard protocols, and there are many kits available for this as well. cDNA can be stored at the -20. It's relatively more stable until it's ready to use. And of course, this should also be in triplicate. So you should have started with 3x samples per condition, and then you're just taking those 3 samples and moving them through this pipeline. Once you have your cDNA ready, most kits will generate cDNA at too high of a concentration to do a good qPCR. And most kits will only generate 20 microliters of cDNA. And that is just not enough to run an entire qPCR plate. So what we normally do is we dilute the cDNA 1 to 10. So if we have 20 microliters, that's going to be 20 microliters of our DNA and 180 microliters of double distilled water. And this is a critical step because you want to make sure that you have enough, and you don't want to overload your qPCR reagents by adding too much DNA into the sample. And then you will need your primers. So before you do any of the actual reactions, you should make sure that you have ordered and reconstituted the PCR primers for the gene that you are interested in. So before we get into actually setting up the reaction, I do want to say that RNA and DNA are very susceptible to RNases and DNases. And these are just naturally occurring molecules that will degrade genomic material. And they're everywhere. They're in our hands, they're in our hair, they're on our benches. They really are everywhere. And so you can get this nice spray called RNase free or RNase away, or you can lay down fresh pads on your bench, or you can alternatively take off any pads on your bench and wipe it down well with ethanol and RNase away. But the important thing is to wear gloves the entire time, to make sure hair is tied back, to try to wipe down any surfaces that you will be using in order to do these extractions so that your RNA and DNA are contaminant free and do well for you in this experiment. So now we've wiped down our bench, we've done our conditions and treatments, and we're ready to start with the first step, which is the mRNA extraction. So in this step, like I said, most people will choose to use the Qiagen kits because they're quite easy to use. But there are other ways to do it that are chemical, like Trizol, which you can look up, or other chemical extraction protocols. But if you do choose to use the Qiagen kit, it comes with these columns, and with the buffers and with a very easy step-by-step protocol. This extraction probably takes an hour to do, and you just follow the steps and you will end up with pretty good, clean mRNA at the end. And you can put in cells or tissues or whatever sample you're interested in. It's compatible with a lot of different samples. So that protocol, since it's so easy to follow and is proprietary to them, is something you can just follow and it will come in the box and it's very easy to do. You will need a microcentrifuge to do it, and you will need Eppendorf tubes, but those are both things that every lab should have. And everything else, all the buffers and columns, come in the box. So now that we have our mRNA, the next step, like we talked about, is to create cDNA. And so this is where we use our reverse transcriptase enzyme. And we already talked about all the components of the reaction. So here we're just going to talk about kits and recipes. So most labs have a kit that they use, and there are many kits available for doing this reaction. So this is just one of them as an example. It's the one that we use in our lab. But like I said, your lab may have a different kit, and the basic idea is the same for all of them. Every kit will have an insert that tells you the exact recipe to use. And so here we're just going to walk through the iScript recipe as an example of how you do this. So if you look on the insert in the iScript box, this is the recipe you will see. It will tell you to mix your RT enzyme with a reaction mix that contains all the other components, so your DTT and your bases and whatever else you need, some water, and your mRNA template. And every kit will give you an amount of mRNA that kit can handle, but I find that using about 2 or 3 microliters usually gets you within this range for any mRNA that you've extracted. So if this is your base recipe, let's talk about how to actually implement this. So first, make sure that you have a thermal cycler available to you, and you have appropriately sized PCR tubes for that cycler. So every cycler is different, but many of them will use tubes like this, which are these very tiny tubes that come in a strip and are called PCR tubes. And with this, you want to be as sterile as you can be. I don't mean sterile like tissue culture sterile, but I just mean sterile in terms of wiping down your surfaces with ethanol and not touching your tips to anything and wearing gloves and trying to avoid that RNase, DNase contamination as much as possible. So once you have those things and you've made sure you have all the reagents available, make sure everything stays on ice the whole time. That's very important for doing this. And then you want to calculate your master mix recipe. And so what I mean by that is that there are 3 components in this reaction that are going to go into every single reaction that we run. So if we're making cDNA from 24 different samples, we don't want to pipette in 4 microliters 24 different times for these samples. So instead of doing that, we just make a master mix that has all 3 components in the correct ratio in one go, and then we can add that master mix to every tube all together. So that is because it allows you to sort of be very consistent. If you make one master mix and vortex it well, then that will be all you need, and every single tube will definitively get the same amount of stuff. So for calculating the master mix, let's say that we have 10 tubes that we want to make. All you will do is multiply all your components by 10, so now you have a 10x mix, and this is the mix that you should make in an Eppendorf tube. So you would simply take this and mix 40 microliters of your reaction mix, 10 of the RT enzyme, and 120 of water in a 1.5 ml Eppendorf tube. And then you should vortex this thoroughly and mix it thoroughly so that you have a very consistent mixture of all the different components. You should always make a little more than you need. So always make 2 or 3 reactions more than you need. So if we actually needed 10, then we should probably make a 12x mix. And that's because you just always lose a little bit in pipetting, and you don't want to end up with less than you need. The other thing to keep in mind is that your mRNA template will, of course, not go into this master mix. And that is because it is different for every single tube. And so the only things that go into this master mix are the ones that will be the same across the board. So you will then place your 3 microliters of mRNA individually into every PCR tube, because remember, this did not go into the master mix. And then you can add your 17 microliters of master mix from here that you made, where you already mixed everything together. So now you can just add it in all together to each tube. And so now you'll have a total volume of 20 microliters. You're then going to run this on the thermal cycler, and every single kit on that insert will also tell you the exact reaction conditions to run it at. Or your lab may have already saved a protocol onto their thermal cycler for qPCR or for mRNA to cDNA. So always check that, always double check against the insert, and whatever the instructions say, put exactly that into your thermal cycler. A nice step to add at the end is a 4-degree infinite hold. And so this can just be a final step that you tack on. And this means that basically it will be held at 4 degrees forever until you come and take the samples out. And this is useful if you're doing multiple things at once, or if you want to start it and leave it overnight. It's the same thing as keeping it in the fridge. And so it'll just be saved for you until you're ready to take it out. So now we've done that reaction and we have the cDNA that we need. So now we're on to doing the actual qPCR. And there are some considerations that are going to go into that qPCR, right? Because the 2 big things we need are cDNA, and then we also need primers. And then, of course, we need cyber and water, but those are things we're buying. So we're buying this, we've made this. So the next thing to understand really well is the primers, because these need to be ordered, and they need to be ordered with certain specifications. So the first thing to understand about primers is primer melting temps, otherwise known as TMs. And these are the temperatures at which 50% of the primer will be annealed and 50% will not be. So if this is your long DNA strand here, then 50% of your primers will be annealed like this, and 50% will be off in the world somewhere. This is an important temperature because if all of your primer is in this position, if your temperature is perhaps too high, then you will never get any amplification. So you need to be sure to check the TM and make sure that it is appropriate. So like we said, the annealing temperature is important, the melting temperature is important because you have to know it for setting up your qPCR properly. And we'll talk about how the temperatures at your qPCR cycling reaction should be adjusted to match the TMs. But for now, understand that your TM on your primer should always be something that you know about and pay attention to. You should try to get melting temps that are similar. So these should be ideally all the same so you can run all your samples together. So if all your primers have the same TM, then you can use the same qPCR conditions to run every single reaction, which is helpful when you want to run a big plate together. So you should really just choose a TM that you like and try to keep everything around that. And then you should always double check your actual annealing temperatures. So sometimes predictors or primer makers, their TMs will not be entirely accurate. And there are other programs that allow you to specifically check the predicted TM. And so you should always go ahead and check that and make sure it actually is what you want it to be. So where do we find primer sequences and how do we manage the TM? There is this tool called Primer Blast, which you can just Google. And it's a great tool for generating good primer sequences. So for this tool, you will enter the NM number from gene cards. So it'll be a number that looks like NM, number, decimal point, number. And this is sort of an ID number for the mRNA sequence. You can find it on gene cards if you just Command F for NM and look for something that should be next to refseq mRNA. So it should say refseq mRNA : NM something. And that is your number that you need. So you put that number in here. So this is the accession number. And then you put in any specifications you have. Most of the time, you don't need to put in too many specifications. This program's pretty good, and it has most things at a reasonable default. Some things that I do find useful to change, though, are the TM. So I sometimes make this 59 and 61 because I want my TM to be really perfectly at 60. And I might also change the number of primers to return, because maybe I only need 2 or 3, so I'll just ask it to return 5 and choose 2 or 3 out of those 5. So you can adjust these and other things as necessary. There are many, many other options on this page, and you can really customize the primers to be anything that you want. But if you're just starting out and you just want a reasonable set of primers, I think those are all the specifications that you really would need to adjust. So let's say that you put in whatever TM you wanted, and you put in the gene you wanted, and you got back a primer sequence. And then you ordered that primer sequence from a primer synthesizing company, and they came to you in these little blue bottles. So now you're like, okay, what do I do with these primers to get them ready for my qPCR? So the first thing to know is that you will always get primers in forward and reverse pairs, and you need both together in one reaction to actually do the amplification. And that is because you have these as your DNA and then these as your primers, and without primers in both directions, you will not get amplification. So always keep the forward and reverse pair of primers together. So they will come in this little blue bottle, blue cap bottle, that is a dry powder format. Before you ever open that bottle, make sure you spin them down. These are very, very tiny amounts of powder. Sometimes they get caught in the cap, and if you open it too quickly, you might just dump it out of the cap without even realizing, and that's the end of your primer. So always, always just give them a brief pulse spin. It doesn't need to be very long. Just sort of pulse it maybe for 10, 20 seconds to make sure that all the powder goes to the bottom where you need it. Then you should find the nanomolarity of the primer. So that will be on the side of the bottle. A typical number is somewhere around 27 to 40 something, and it'll say nanomoles next to it. You're going to multiply that by 10, and then you're going to add that many microliters of water into that primer tube. So remember, that primer tube has some invisible white powder in it that's hopefully at the bottom now that you've spun it down. And now you're adding this water directly into that tube so that it can reconstitute whatever is at the bottom and dissolve it properly. So we take this nanomolarity, we multiply it by 10, so let's say it's 27. So now we're going to get 270 microliters, and we're going to pipette that and reconstitute. This will create a 100 micromolar master stock that you will then use for everything moving forward. You should vortex this very well before moving forward with anything else. So once you have done that, your next step will be to create your actual working stock. So here you'll create a 10 micromolar working stock. This will be in a fresh tube, not the same tube that you used, right? Because you want to save this and keep it. And then use a fresh stock that will contain the stock that you actually use in your qPCR. And you will dilute 1 to 10 from your master mix. So the best way to do this is to create a forward and a reverse primer mix. So that means that you have a 100 micromolar master stock for your forward primer and a 100 micromolar master stock for your reverse primer. And these have to go together in your reaction. And it can be really easy to get confused about which primers go together. So the way to kind of save yourself from that is to just make it as one stock, as one working stock, and then you can just use that stock for all of your reactions and not worry that you might have mixed up the primers. So to do that, since it's a 10 micromolar working stock, you dilute each one 1 to 10. So you would do 10 microliters of your forward primer and 10 of your reverse, and then fill it up with water. So total volume here is 100, and each of these is 1 to 10 in that 100. So 10 of forward, 10 of reverse, fill the rest with water. And this is the stock in the Eppendorf that you will actually use for all of your qPCR reactions. This stock you will store, and you can use it to keep replenishing your working stock and to have as backup in case something goes wrong. So as I said, if it has both components mixed in, it's just a tip. It will help it make it easier to make sure you have the correct combos. But you could also just make 1 to 10 stocks of each forward and reverse separately if you prefer to do that. So now let's plan out our plate. We have our primers and we're ready to actually do this qPCR. And like every experiment, the first step is always going to be to plan very well. So for qPCR, you will typically run it in a 96 well plate. And because 96 well plates have lots of wells, it's a good idea to write down your entire plan before you start pipetting. So the important thing to realize is that for qPCR, you need a lot of replicates to make sure that your samples are actually good and your results are good. Because any small deviation in your replicates really throws off this assay. So for every sample and every gene, so every combination of every sample and gene, which is the same thing as saying for every combination of every sample and primer, you should have biological triplicates. These are the triplicates that we made. We've been carrying triplicates through. And you should have technical duplicates. This is for pipetting error. So if you don't have true biological triplicates, then you should at least do technical triplicates, if nothing else. And you have to remember that GAP-DH, just like with Western blots, we use beta-actin as our normalizing control. Here, GAP-DH or 18S is another common one. Those are your normalizing primers. So those have to be included in every single plate that you run. Just like we include actin on every single blot we run, we have to include a control primer in every plate that we run. So what this looks like is, let's say that I have a sample I'm interested in. So let's say it's a DMSO GBM43 sample, and I have a gene I'm interested in, P53. Then for this set, for every DMSO 43 that I want to look at P53 in, I need 3 biological replicates and 2 technical replicates. So I need 6 wells in total where I will be putting this sample in. And this will be the first replicate of that sample, the second, and the third. And then this row will be the exact same thing again because it's our technical duplicate. So make sure that makes sense to you before you move on from this slide, because it's very important to set this up correctly. So for every sample and every primer combo, this is what you need to do. So of course, since we need GAP-DH in every plate, we would do exactly this with GAP-DH as well. So let's practice that for just a second. So let's say that we are interested in looking at P53 and EGFR in DMSO and TMZ. What I find most helpful is to build this as one section. So build one section of your plate that will go to one primer, and then you can repeat that for every primer you're interested in. So let's just start with looking at our samples. So if we build one section, then we have DMSO1, DMSO2, and DMSO3. Those are our biological triplicates. And then, like I said, we're going to do the exact same thing again to have that technical duplicate. So we will do D1, D2, and D3 one more time in order to have that duplicate. And then we have a second sample. So we've done our first sample. Now we have a second sample that we're interested in, which is the TMZ. So we'll do the same thing again, biological triplicates, and then the technical duplicate. So these are our 2 samples. So we have a control and a treatment. So this is one block. This is what we need to include for one primer. So now we can just repeat this block again over here and again over here. And for this, we'll add our P53 primer, and here we'll add EGFR, and here we'll add GAP-DH. And these wells are going to be just exactly the same thing again and again, and the rest of this plate will be empty. And that will be our scheme. So it's important to write this down because when you pipette, everything will just look like clear liquid, and you will not know what is what. So it's good to write it down and have it as a reference, even when you're pipetting, to be able to check, okay, like I'm at well 2B, what do I need to put in well 2B to make sure that you don't pipette wrong or get off somehow. So, and then make sure you understand the concept of having these triplicates and duplicates and setting up one primer set first and then just copying that over for every primer set or every gene that you want to examine. We'll try one more example, and this is for the case where you may not have remembered to make biological triplicates. Now, of course, this is non-ideal, and you should always try to have triplicates. But in the event that you didn't, for whatever reason, I just want to show you how you might still set up that plate and try to understand a pilot study of what might be going on until you make those triplicates. So let's say that you have DMSO and TMZ again. Those are your two samples. And we're going to set up for one block, just like last time, so one primer set. So for one primer set, we have two samples, and this time we don't have biological triplicates, so we're just going to do technical triplicates. So we're going to do the same sample 3 times, technical triplicate. And then the next sample, again, 3 times. And that's our technical triplicate. So this allows us to still analyze and somewhat understand what might be happening. And this is our first block. We don't have 2 rows this time because we don't have biological triplicates. We've already done our technical replicate. And then we're just going to make 3 blocks for 3 primers. So this will be P53, this will be EGFR, and this will be GAPDH. And remember, we need 3 blocks because we cannot add more than 1 primer into one well. In other words, it has to be 1 primer per well, because remember, the primer is just going and amplifying DNA, and then that amplification is being measured in fluorescence. So if we added more than 1 primer, then more than one thing would amplify, but we would not know which was which. So remember, 1 primer per well per sample. So this is the actual process once you've gotten to the point where everything is prepared. So at this point, you've figured out your primers, you have a complete cDNA sample, and you have your cyber, and then you have the plates and reagents that you need. So for qPCR, you need a specific plate that is compatible with your machine. So this is the one that we use, but there may be others. Make sure it's compatible with the machine you're using. And you need a special film to cover that plate. So for this particular plate, it's this yellow film, but that can also change depending on what plate you're using. So make sure you have the appropriate reagents ready. Now you will have planned your plate. Remember, this is the first and most important step, to plan your plate well and write it down so that you can pipette it properly. You can underestimate how easy it is to get mixed up, so don't think that you can do it without writing it down. Always write it down. Then you're going to go back to that same concept of making a master mix. So you would do what you did with the cDNA, where you think about how many wells you need for each primer. So here you have to make a master mix for every single primer, because remember, the primers are changing. So for each block of wells, you're using a different primer. So you need to make a master mix for each primer or gene you want to look at. So for example, in our previous ones where we were doing P53, EGFR, and GAPDH, we would have made 3 mixes, 1 for each primer. And you will calculate from the recipe. So this is the 1x recipe. So in our previous example, we had approximately 12 wells filled. And so we would make 14x because we need to make a little bit extra because we will lose some in pipetting. So we'll make a 14x recipe. So then we just multiply these values and then add all this together to make our master mix for the primer of interest. And then, of course, we don't add our cDNA because this will change from well to well. And so we add that at the very end. So your master mix only includes these 3 things. Once you have your master mixes ready, always vortex. Vortex everything to mix it really well, because the whole point of making a master mix is that you have a very consistent mixture and you can trust that every well will be the same. But if you don't vortex, then it won't be well mixed, and that will no longer be true. So you vortex, and then you go ahead and you add 1 microliter of DNA to every well, following your written layout. So following along exactly with what you have written down, 1 microliter for each well based on the sample that you wrote down that should go into that well. And because 1 microliter is such a small amount, you do want to make sure that it actually gets into the well. And the best way to do that is to angle your tip and put it against the wall of the well about halfway down. and let it go. And you should see the droplet form on the wall, and you should be able to see droplets in every single well as you pipette. And that will allow you to check at the end that you actually got your cDNA into every single well appropriately. It's also a good idea to change tips constantly for this because you do not want to contaminate samples against each other. And you also are pipetting just such a small volume that having a fresh tip every time helps make sure that your pipetting stays really accurate. Once you've done that 1 microliter, you take your master mixes. Again, remember the appropriate master mix for the primer that is supposed to go into that block. And you add 9 microliters of your master mix into the well. And again, make sure it goes into the well, although this is a larger volume, so it's relatively easier to make sure. You will then seal the plate with the special film that we talked about, and you will spin it down very briefly to remove any bubbles and mix. At the end, you should see all liquid at the bottom of each well, and it should look very consistent. And then you will run it at whatever appropriate conditions that we'll talk about in a second. The one point I want to make is that here I have provided you with a 10 microliter PCR recipe. The standard is to do 20 microliters, in which case you would simply double every component that I've listed here. But 10 microliters can be used if you're trying to save cybergreen because it can be quite expensive. So let's just go through an example of how we might do this. So again, we'll go back to our qPCR for P53 and EGFR, and of course GAPDH as our control. And we're gonna run it in those DMSO and TMZ samples we already talked about. So we already designed this plate, and here I've just written it out for you in a table so that you can see it. You can see we have our technical duplicates and biological triplicates, and then we have one set for every single primer that we're interested in. So how many master mixes do I need to make? We already kind of talked about this. We need 1 for every single primer, so you would make 3. How many reactions should each one contain? So if we count the number of wells we need for each primer, it's 12. And we always want to make 2 to 3 reactions extra. So I would make 14 to 15x of each master mix, each of 3 master mixes. So you will make this 3 times at 14x, whatever is listed here, switching the primers every time to the different primer. So you're making a master mix for each primer. Given the 1x recipe, what exactly is the recipe for each master mix? So this will simply be x14, each of these. And what will you do after you've made the master mix is what's on the previous slide. So you would go ahead and add your DNA, add your master mix on top, cover it with a film, spin it down, and then put it in the machine to run. So now let's talk about how to actually run the qPCR reaction. So this is a pretty standard thing, and these are the usual setup that you can do, but most labs already have this set up on their qPCR machine, so you can just choose the protocol and use it. The only important thing to note here is that we go through all the usual steps. So it denatures, then the primers anneal, then they extend, and then you have 2 strands of DNA, and then you go back through your cycle. So you do this 40 times, like I said, to get the right amount of DNA and check the amplification. 2 things to note. One is that we talked about Tm. So Tm, remember, was the melting temperature. And Tm is the temperature where your primers are basically half of them are not working. So you definitely don't want your annealing temperature to be higher than your Tm. Because if it were, then your primers would not be annealing, and so you wouldn't get extension or amplification. So a good rule of thumb is that the annealing temperature should always be 3 to 5 degrees under the Tm of your primers. And of course, that depends on your primers all having a similar Tm. Another thing you can do to make sure your primers are actually working and giving you one unique product is to add a melt curve after 40 cycles. And a lot of machines allow this. It's just a simple curve that goes across every temperature. And it allows you to see how your primers are doing over time. So you should see one peak, which corresponds to one product. And you should also see clean melt curves for your primers, which show that it is working the way you would expect. But the most important takeaway here is that this annealing temp has to be correct. If it's not, then you will not see amplification and your qPCR will not work. So always check your TMs, make sure they're the same across your primers, and then set an annealing temp appropriately. So once you run your qPCR, you'll go back to the machine and you will see these curves. And so this curve is basically an amplification curve. So for every well, you will get a different amplification curve. And the machine will calculate a CQ or CT value for you based on where this curve crosses your threshold. So for example, this one would be probably 11, and this one would be something like 22. The lower the CT is, or CQ is, the higher your expression is of that gene. So 11 is very, very high expression. Normally our housekeeping genes like GAPDH and 18S, which are sort of our normalizing genes, show up around 16 to 18. And other genes will show up greater than 20. If something has a CT greater than 35, that's basically water. So at that point, your gene expression is so low that it might as well not be there, or your sample just does not have any cDNA in it. So like we said, CT is where it crosses the threshold. Sometimes you'll have to set your threshold. Some programs will set it for you based on all of your samples. We talked about the normal values. And then you do want to check your amplifications thoroughly. So amplifications for one sample, for the 3 replicates or 6 replicates of that sample, should all look very, very similar. So like you can see these green curves clustered here or these red ones clustered here. They're basically so on top of each other that it's almost hard to see that they're separate curves. And that's what it should look like. You should see very, very little variation in your replicates. If you see that I have a green curve here and then I have another green curve that's like way off here, then you know that this one isn't real. Something went wrong with this sample. And that is a reasonable justification to ignore it. Or if that's happening to a lot of your samples, then you will need to just rerun your samples. And then a good rule of thumb for checking that you have good replicates is that they should be no more than 1 point apart. That's like the absolute max. Ideally no more than 0.5 apart, or like I said, basically on top of each other. The other curve you'll get if you run it is the melt curve. And this is really useful for seeing how many products a primer is giving you. So a good primer should look like this. It should give you one clear peak. There's actually 3 curves here, and they're so tightly overlapped that it's hard to tell, which again is a good thing that your replicates are so tight. And this tells you that I am getting one clear product at one defined set of temperatures from my sample. This is less good, especially if it looks like 2 even peaks. That's even worse because now you're getting 2 products and you can't be sure that it is actually the correct gene that you're supposed to be interested in, because your primer should have been designed so it's specific to one gene and would only give one product. So this kind of thing requires more investigation. Sometimes it is true that it's still measuring the gene correctly, but in general, you would hope to see just one clear peak because that is sort of the reliable result, and then you can move on and analyze the data. It's also a good way to make sure that your primers are working. So if you see a lot of peaks, then you know that you probably need to optimize your TM or reorder new primers. So it's a good way to check quality. So a few notes on troubleshooting just to wrap up the experimental section. If your primers don't work, we kind of already talked about this. If the amplifications or melt curves are bad, the first step always is to perform a temperature optimization. Because like I said, the annealing temps that are given to you or the melting temps that are given to you are not always accurate. So sometimes it will tell you the TM of this primer is 60, but it's actually like 68. And that's why your PCR did not work. So always perform a temperature optimization, and that means that you take a plate that all has the same samples, every single well is the same, and you run it so that every row is a different temperature. So for example, this could range from 50 to 70, and then you have all the temperatures in between. And that will allow you to know whether or not you have the right annealing temp. So this will change the annealing temps, and then you can look for where the melt curves and amplifications are the best and the most aligned. and that's the temperature you should use moving forward. Another key thing to point out is that bad pipetting is probably the most common issue with qPCR. So if your replicates look bad, if they're not quite the same, it's probably a pipetting issue, and so just be really, really careful when you pipette. And then two other things that might be going wrong that are less common are making sure there's no RNase contamination, and then also making sure that your gene is expressed in the sample and that everything is working as expected. And GAPDH should be a good control for that to make sure that everything is otherwise working. So to wrap things up, let's talk quickly about how to process this data, sort of how you'd present it, interpret it, what you would do with it. So the first thing to understand is how to analyze qPCR. So basically, when you run a qPCR experiment, what it will spit out is a bunch of samples, the primer that you used, and a bunch of CT values. And you have to turn these CT values into expression values. So let's talk about how exactly to do that. So let's say that you have your 3 samples. So this is, DMSO, replicate 1, 2, and 3. And this is for your GAPDH primer. And then you have DMSO 1, 2, and 3, and this is for your P53 primer. And now you want to calculate, so this was CT values, right? And you want to calculate expression. So first, let's put in some reasonable CT values. So GAPDH is usually somewhere around 18. So we'll just put 18 for all of these, somewhere around 18. And your other genes will usually be somewhere in the 20s, hopefully. Remember, over 35 is basically you don't have any expression. So let's put 24.5 here, maybe 24, maybe 24.5. And you'll notice that these are all less than 1 point apart, that the replicates are really, really tight, as they should be. So once you have this column, your next thing is going to be to generate a column that is the difference, right? Because you want to normalize all of your genes against GAPDH. So this is called the delta CT, or the change. And you want to basically take every gene and subtract the GAPDH from it to really normalize it against GAPDH. So this would be 6, 6, and 6, the way I've done the math. But in reality, you might get something more like 6.2, 6.1, 6.5. Hopefully they're not too far apart, assuming that your originals are not too far apart. Then you will go on and you will next calculate the power, right? So what we just did was this. We did it with GAPDH, not 18S, but 18S is another housekeeping gene you could have used. Now we're going to calculate the power. And so we're not actually doing this step where you calculate a double delta, because we are going to just go first and calculate the relative expression and then think about the double delta. So in this case, for power, it's always going to be 2 to the negative delta CT. So in this case, it's going to be whatever 2 to the negative 6.2 is. And the negative is really, really important because that's how you get a decimal number, which is what you would expect to get. So always remember 2 to the negative, whatever that CT is. That is your power value, and this is basically your measure of expression. Now, if we had a control versus a knockdown that we were specifically interested in, like if I had my TMZ sample here as well, I could have done this double delta calculation. But I personally prefer to just graph all the conditions and show the control on my graph. And if I did this, I would basically eliminate the control and only have my treatment condition. So I am not going to do this. I'm going to keep them separate. So I would have 3 numbers for DMSO and then 3 more for TMZ. And this is what you can put into Prism to graph. But remember that we talked about how qPCR is a relative tool. And it is not very helpful to tell someone that, my DMSO expression is 0.05 and my TMZ expression is 0.005. That's really hard to interpret. But what might be helpful is to normalize this so that it's 1, and then this becomes 0.1. And so now everyone can clearly see that this is 1/10 of this. Or alternatively, what you normally see is more like this is 1 and maybe this is 3 or 5 or 2. And so then you can say, I have a 2-fold change in my treatment over my control. That's really the kind of statement you want to be able to make. You don't want to say things like my control is 0.05 and my treatment is 0.002. That's confusing and hard to interpret. So in order to do that, we have to do one more step where we normalize everything. So in order to normalize, we're going to do one more step where basically we're going to start this time with our power values. And we're going to say that this is DMSO and this is TMZ. So obviously DMSO is our control here. And let's say that our power values were something simple like 0.1, 0.2, 0.3. And our TMZ values were also pretty simple. Maybe we have a 3 and a 1 and a 2. This is probably not what you'll have, but we'll just, for the sake of argument. So what we want to do is make the average of DMSO 1. So we want to calculate the average, the current average of DMSO, which should be 0.2. And now we're just going to take our normalized power. So this is normalized power. And it's going to be the power divided by the normalization factor, which is the average of DMSO. Because if you divide all of these by its own average, you should get an average of 1. So this will be like 1.5, this will be 0.5, and this will be 1, which averages to 1. And then we're going to divide all of this by 0.2, so we're going to get 15, and we're going to get 20, and we're going to get, we're going to get 10. So, or something like that. Basically, we divided all of these by 0.2. So now we can clearly see that if the average here is 1 and the average here is 15, that we have a 15-fold change over control. So this is just a lot easier to see. And so I will often calculate this normalization factor and then put these normalized power values into Prism to graph so that I have a really clean graph that looks like this, where it's like 1, 5, 10, 15, and this is my control, and then this is my treatment. So it's a really easy axis for people to read and interpret, whereas like it would be much harder to interpret if it was a bunch of decimals here. Another key thing to realize is that this exponential is the reason that any pipetting error will cause a lot of issues, because you're taking this negative and you're taking a delta. And so if there are any pipetting errors that even set your different samples off by more than 1 point, that's going to get magnified to like a 2x or 4x change. And that's going to create a lot of issues for error bars in calculating significance. So your pipetting needs to be really, really tight to get good results out of qPCR. All right, so interpretation we've already talked about to some degree, but the important thing to realize is that this is a relative expression. So it's not telling you absolute mRNA expression. It's telling you compared to your control and normalized to GAP-DH, what is a relative expression of any given gene. And so, for example, here you can see that this is a 3x fold change compared to its control. Or maybe this is a 2x fold change compared to its control. So that's how you should interpret and present it, as like a relative fold change over a given control. And then in terms of presentation, all the usual things apply. So everything should be very well labeled. Always call your y-axis a relative axis. Make sure any conditions are along the x-axis. And always do error bars in statistics. You should do this for any experiment, but for qPCR especially, it's somewhat meaningless without the error bars and the statistics associated with it. And as a quick note, if you are not aware of this, Prism is the software that's usually used for scientific graphing. And so all of these graphs can be made in Prism, and the error bars and stats are very easily calculated in Prism. And so it's a great thing to download if you don't have it already. So in this video, we covered the basics of qPCR, but the theory behind how we do PCR and the practical applications of doing it in the lab step by step. I hope this was helpful, and I hope the analysis interpretation sections will also help inform how you interpret your qPCR. Thank you so much for listening. If you want to follow the lab, This is our website and this is our Twitter handle. We also have a YouTube channel with these videos and many more. And then if you want to see more of these videos or more information like this, or if you have any feedback, please feel free to contact me or the lab. Thank you.